Hello everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode we're going to discuss the convergent evolution of electroreception. So let's jump right in. At 23 episodes in, we are finally at the end of the mammals, collecting everyone together 180 million years ago in the Toarcian epoch of the early Jurassic. We have left the therians behind, the marsupials and placental mammals, and find ourselves among a collection of stem therians: Dryolestida, Spelacotheroidea, Multituberculata, and Eutriconodonta. The Dryolestids are a collection of small insectivorous mammals that existed from the Middle Jurassic to the early Cretaceous. Because most mammals at this time were small insectivores, there has been some debate over the members of this clade. At the very least, the families Dryolestidae, which includes Dryolestes, and the possibly paraphyletic Parodontidae, which includes Parodon, are in this clade. However, whether Meridiolestida, which includes the late surviving Necrolestes, is also included is controversial. Interestingly, the tooth morphology of Parodon valens is strongly suggestive of a mostly earthworm diet, being highly morphologically similar to the extant golden mole Amblysomus. As for Necrolestes, it too was specialized for a subterranean life. As a 2007 paper points out, quote, "One of the most conspicuous features of the Necrolestes forearm is the medially curved olecranon process of the ulna. This feature is present to an even greater degree in the Palaenodonts, Xenocranium, and Epoicotherium. Golden moles, Nodorictes, and Euphracis show a similarly curved ulnar olecranon." Close quote. Next, Spelacotheroidea was previously grouped under the clade Symmetrodonta, but this clade seems to be either paraphyletic or polyphyletic, causing it to fall out of usage. Spelacotheroidea existed from the early to late Cretaceous. A complete skeleton of the early Cretaceous Zangiotherium shows that these mammals had venom spurs, like modern monotremes, indicating that this is a characteristic shared by all early mammals. Third, Multituberculata existed from the Middle Jurassic to the late Eocene, and their ecological niches were similar to the other mammals mentioned so far. Interestingly, in this clade, Phyllocomys from the late Cretaceous of Montana is the earliest evidence of multi-generational nesting behavior in mammals. There have been vigorous debates over why and how the multituberculates went extinct, with some researchers proposing they declined as a result of competition with rodents and other placental mammals. However, this has yet to be substantiated. The final group of stem therians we're going to look at is Eutriconodonta. This group is highly diverse in both morphology and ecology. While some of these, like Yanoconodon, look fairly similar to the other insectivorous mammals we've been discussing, other Eutriconodonts took very different evolutionary paths. For example, Velaticotherium from the Middle Jurassic of Asia appears to have been adept at gliding, and its South American relative, Argentoconodon, likely was too. Now that we have gathered all the stem and crown therians together, we can call them by their total clade name, Boreosphenida. Remember from the hippo's tale that Boreoeutheria was the northern true mammals, and Boreosphenida is similar in etymology. Boreo here still means northern, referring to North America, Europe, and Asia, but sphenida refers to the shape of the molars. Traditionally, therians and their extinct cousins were called Tribosphenida because of their triangular or V-shaped molars, but such molars also exist in the sister clade of Boreosphenida, Australosphenida, the monotremes and their extinct relatives. Australo means southern, referring to Gondwana, not Australia. As with the other splits we've discussed recently, the main morphological difference between Boreosphenida and Australosphenida is the relative shapes of their teeth. Importantly, Australosphenida has a ridge that runs along the front of the molar, while Boreosphenida doesn't. There isn't much else. As a result, there has been a lot of debate about where and when these clades arose. Previously, researchers have considered the families Ausktribosphenidea, Bishopidae, and Henospheridae to be stem monotremes, but a 2022 study lumped these groups into Boreosphenida. If this is the case, then actually both Australosphenida and Boreosphenida originated in Gondwana, with the latter group radiating into Laurasia later. But that also means the fossil record of stem monotremes doesn't begin until the early Cretaceous rather than the Middle Jurassic. Of course, we come back to the issue of physiology and ontogeny not being preserved in the fossil record. We know that extant monotremes, the platypus and echidnas, lay soft, leathery eggs, but marsupials and placentals don't. When did this changeover occur? Our common ancestor with the egg-laying, or oviparous, monotremes lived 180 million years ago, and our common ancestor with the live-bearing, or viviparous, marsupials lived 160 million years ago. That gives us a window of just 20 million years for the switch to occur. That might sound short, but in fact, there are amniotes alive right now that are capable of both oviparity and viviparity, such as the European common lizard Zootoca vivipara. Within this species, it ancestrally evolved viviparity, and then populations in northern Spain and northern Italy independently re-evolved oviparity. Interestingly, the Italian population gives birth to offspring with thicker eggshells who are less developed at the time of oviposition compared to the Spanish population, possibly indicating that the Italian population evolved oviposition before the Spanish population. Furthermore, according to a 2006 paper, quote, most oviparous squamates retain their eggs in the uterus for periods that, depending on the species, represent 20% to 80% of the total embryonic developmental time. Hence, the emergence of viviparity in squamates may be viewed as an endpoint along an egg retention continuum and not as a discrete novelty requiring dramatic character changes. For example, many species of viviparous squamate retain a thin eggshell membrane enveloping the embryo during gestation. Similarly, as in oviparous species, most viviparous species of squamate still exhibit lecithotrophic, from the yolk, embryonic nutrition. These observations indicate that the characteristics essential for oviparity may not be irremediably lost in many viviparous squamates, close quote. And the fact that viviparity has evolved over 100 times in squamates seems to suggest that the transition is relatively easy. It's also worthwhile to point out that squamates and mammals share the same extra embryonic characters such as the amnion, allantois, chorion, and yolk sac. Thus, even the placenta itself is formed from the same tissues in squamates and mammals. A 2014 paper points out that the uterine plasma membrane transformations, similar to those that facilitate implantation in the rat, occur during pregnancy in skinks. Additionally, the transport of nutrients to the developing offspring is simply a matter of changing the expression of existing genes and tissues rather than the wholesale invention of new ones. For example, quote, in oviparous species, calcium ATPase is upregulated in the uterus only during eggshell deposition, but is upregulated throughout pregnancy in the viviparous skink, Pseudomoia spenceri, presumably to provide calcium to the embryo throughout development, close quote. A 2017 paper found that placenta formation in the skink Mabuya involves a refunctionalized retrovirus similar to gamma retrovirus, and the gene derived from it is called sensitine MAB1. The protein produced by this gene is called sensitine, and it has popped up independently in mammals due to infections by other retroviruses. Sensitine facilitates the fusion of multiple cells into a single cell called a syncytia, which is necessary for placenta formation. In catarrhine primates, the retroviral-derived gene ERVW-1 is involved in fusing cytotrophoblast cells to the syncytiotrophoblast. All of this to say that the transition from oviparity to viviparity was probably not a difficult one. Now we come to Australosphenida, which apparently only includes the monotremes and a handful of extinct cousins. Monotremes are different from marsupials and placentals not just in egg-laying, but also in having a single exit for both their digestive and urinary tracts, called the cloaca, just like birds and reptiles. Monotremes have an interclavicle bone near the shoulder, like reptiles, and their cochlea is nearly straight, like reptiles, unlike the coiled cochlea of extant therians. However, monotremes have a single lower jaw bone, the dentary, like all other mammals. The transition from jaw bones to inner ear bones is a story we'll tell next time, but it's worth noting that monotreme ears are much nearer to the jaw than those of other therians. Monotremes also have hair and secrete milk, just like all other mammals. Monotreme milk isn't secreted from a nipple, though, but a milk pad. However, this appears to be a unique character of monotremes, not an ancestral character for mammals. The oldest stem monotremes come from the early Cretaceous of Australia, such as Teinolophos, Steropodon, Collecodon, and Cryorichtes. This might seem to suggest an Australian origin except for Obdurodon sudamericanum, which lived in Patagonia from 63 to 61 million years ago. This monotreme, along with lungfish and myobatrachid frogs, very similar to those in Australia, indicate faunal similarities between South America and Australia as recently as at least the early Paleocene. A 2009 study puts the common ancestor of echidnas and the platypus at between 19 to 48 million years ago, so the fact that the platypus like Steropodon existed before that means that echidnas evolved from semi-aquatic platypus-like ancestors. The reason for such a large range of dates is due in part to there being such a fragmentary monotreme fossil record and having only a few living species. In fact, there is only one extant species of platypus which is native to Western Australia, and 4 extant species of echidnas within the genera Tachyglossus and Zaglossus native to Australia and New Guinea. There was formerly a giant echidna called Murrayglossus from the Pleistocene that reached over 3 feet in length. Echidnas, as mentioned previously, are terrestrial insectivores covered in hair and keratinous spines. They are known to eat termites and ants, earning them the name spiny anteaters, even though they are quite distantly related to the true anteaters. Thus we come to the platypus, who is aquatic, possessing webbed fingers and a paddle-shaped tail. Early accounts by naturalists of the platypus were originally written off as hoaxes, and apparently some people today still think the platypus is an amalgamation of different animals. But no, the reptilian characteristics of monotremes aren't chimeric additions, but ancestral characters that earlier mammals possessed, which were lost in the more derived therians. One characteristic in particular seems to confuse a lot of people: the platypus's bill. Indeed, it is often referred to as the duck-billed platypus, even though the platypus's bill is very different from that of a duck's. Whereas the duck's bill is actually 2 halves, the platypus is one whole structure. Additionally, the platypus's bill is soft, not hard like a duck's, and is full of sense receptors where the duck's isn't. This is the basis for the duckbill's tail. The platypus forages for invertebrates underwater, and decades of study on this mammal have revealed that it essentially never opens its tiny eyes underwater except for just before surfacing. Nostrils and ears are also closed underwater. Instead, the platypus's main apparatus for detecting aquatic invertebrates is the electroreceptors in its bill. Echidnas too have electroreceptors. The short-beaked echidna has the fewest, while the long-beaked echidna is intermediate in number between the short-beaked echidna and the platypus. The platypus has 100,000 total sense receptors in its bill, of which there are 2 types: 60,000 push-rod mechanoreceptors and 40,000 mucus gland electroreceptors. The electroreceptors are arranged in stripes along the top and bottom of the bill, but are markedly concentrated at the edges of the bill. The mechanoreceptors are viewable externally as microscopic domes covering the bill. Inside the dome is the push rod, so called because they are indeed tiny epidermal rods that are free along their length, being only attached at the bottom. As such, these rods can rotate somewhat around their attachment site. At the base are sensory organs resembling Merkel cells and Pacinian corpuscles, both of which are mechanoreceptors widely distributed among mammals. The platypus's mechanoreceptors are evidently only useful underwater. As for the electroreceptors, these are modified mucus glands, and at the base is a radial arrangement of about a dozen free nerve endings very close to, but not quite in, the mucus duct itself. When animals use their muscles, very weak electric fields are produced, and the platypus picks these up with its electroreceptors. Evidently, these structures are sensitive enough to respond to 50 microvolts per centimeter, which would be useful for detecting small shrimp. When the platypus detects electrical impulses of prey, it begins swinging its bill side to side, a motion called a saccade. The electroreceptors indicate the direction of prey, and the lag time resulting from the faster action of the electroreceptors relative to the mechanoreceptors might indicate the distance of the prey. This is sort of like how we can estimate the distance of a lightning strike from how long it takes to hear the accompanying thunder. Electroreception only works underwater because the resistivity of air is 20 billion times higher. That's far too high to transmit biologically generated, detectable, and relevant electric signals. Side note, in a 1998 paper, researchers identified how much of the platypus's neocortex is dedicated to its bill, and it turns out that it's a lot. Just for comparison, the human neocortex is largely concerned with the hands and face, especially the parts related to chewing and speaking. In the platypus, the sense receptors input to nerves, like the trigeminal nerve, which is very large in the platypus, and the sensory inputs are combined in a structure resembling the ocular dominance columns of primates. For reference, ocular dominance columns are stripes of neurons in the visual cortex that respond preferentially to input from either eye. And interestingly, the electroreceptors actually form in juvenile platypuses before the mechanoreceptors. But the platypus isn't the only mammal with a face full of electroreceptors. In 2012, researchers in Germany demonstrated the electroreceptive capabilities of the Guyana dolphin. This dolphin has a very different system compared to the platypus. For starters, the electroreceptors are evolutionarily derived from mechanoreceptors. Vibrissae, or whiskers, are mechanoreceptors that nearly all mammals have, and toothed whales, or odontocetes, are born with vibrissae that atrophy shortly after birth. Though the hair itself is gone, the leftover crypt functions in electroreception. Odontocetes form a glycoprotein-based gel during skin renewal, and this is permanently embedded in the nanoridges of the epidermis. Evidently, this gel is highly electrically conductive, and combining this with the crypts being densely innervated by branches of the trigeminal nerve seem to facilitate the transduction process. The nerves are much closer to the crypts in the Guyana dolphin than in any other mammals. More data is definitely needed, though. What about outside mammals? The paddlefish, of which the only remaining species is sadly the American Polyodon spatula, has a very long extension of its cranium that is packed with electroreceptors too. And just like in the platypus, these electroreceptors are primarily concentrated along the edges of the paddle. However, unlike the dozen sensory stripes on the platypus's bill, the paddlefish has its electroreceptors concentrated in just 2 stripes. Similar to the platypus, the paddlefish has very little use for its eyes, which are quite small, but unlike the platypus, the paddlefish consumes zooplankton by filter feeding. Paddlefish have some 75,000 electroreceptors in their paddle, which are extremely different morphologically compared to the sensors of the platypus. The paddlefish's electroreceptors, ampullae of Lorenzini, are homologous to those of sharks and rays. Indeed, the ampullae evolved early in jawed vertebrates and are also retained by cartilaginous fish, coelacanths, lungfish, amphibians, bichirs, and sturgeons. They have been lost in amniotes and teleosts. The ampullae are pores leading to a pear-shaped space, the bottom half of which is lined with ciliated sensory epithelium. It's quite different from the free nerve endings of the platypus's mucus glands. What's also quite different is that evidently paddlefish only need their paddles while juveniles. As juveniles, paddlefish catch individual planktonic organisms, like adult platypuses, but as they grow older, they switch to ram feeding like baleen whales or the basking shark. The ampullae do probably play a minor role in mechanoreception, but the extent to which is currently unknown. However, because the paddlefish is far more electrosensitive than the platypus, this seems to downplay the role of mechanoreception. The paddlefish are so sensitive that they are unlikely to pass through the partially open gates of locks during migratory events because of the unnatural electric field produced. Among arthropods, there is a single known example with probable electroreception, the Ordovician trilobite rhedocalamine. This trilobite looks pretty average overall, except for its long rostrum, which appears to have been studded with sense receptors. All the animals we've discussed so far participate in passive electroreception, where they merely receive electric signals. But some teleosts perform active electroreception, also known as electrolocation, where they generate very weak electric fields. This is the case for the gymnotiformes, or neotropical knifefish, of South America, and Mormiroidea, the elephant fish in their kin, of Africa. Though, as we established earlier, animals generate very tiny electric fields from nerve and muscle tissue, these fish organize their muscles into blocks that generate a much stronger field. These specialized muscle cells are termed electrocytes because they shunt large volumes of sodium ions across their membranes. Some fish, like the electric ray torpedo, electric catfish, the stargazer Astroscopus, and the gymnotid electric eel, generate such large electric potentials that they can even stun or kill prey. These fish also have electroreceptors, which, just like in the paddlefish, are modified mechanoreceptors. Fish have a lateral line system that runs along their face and sides, and it consists of a subdermal fluid-filled canal lined with structures called neuromasts. These neuromasts have hair cells that respond to mechanical changes in water. In Gymnotiformes and Mormiroidea, independently, modifications on these structures have converted them into electroreceptors. When the mormirid Gymnarchus, or the unrelated gymnotid Gymnotus, swims, it holds its body rigid to receive signals from the electroreceptors, so swimming is nearly entirely accomplished with a single lateral fin. Interestingly, Gymnarchus has its fin on top, while Gymnotus and the other knifefish have their fin on the underside. To finish off this tale, there is one honorable mention, the star-nosed mole Condylura cristata. This is a semi-aquatic mole native to northern North America, but unlike the animals mentioned so far, it completely lacks electroreception. Its special appendage, the 22 tentacles around its nose, are extremely sensitive to touch, being covered with over 25,000 epidermal sensors called Eimer's organs. Unsurprisingly, the star-nosed mole's neocortex is predominantly concerned with both the tentacles and the front claws. And comparative studies of other moles, such as the coast mole Scapanus orarius, have a proto-star consisting of backward-facing short modules of sensory organs attached to the sides of their snout. So, that's the platypus's tail. Electroreception has evolved repeatedly in vertebrates through the exaptation of various existing systems. So, thanks for watching, and I'll see you next time.